Hello, my name is Trace, and this is my presentation, Kombucha Leather Durability, Sugar Concentration's Effect on Bacterial Cellulose, abbreviated KILD SCOBC. My advisor was Dr. Hatley. First, I'm going to start out by introducing some key terms. Kombucha is a probiotic, acidic, non-alcoholic fermented tea beverage that originated in the Asian continent. And the process that I looked at here was the formation of the cellulose pellicle that grows on the surface of the liquid. This is sometimes referred to as a SCOBY, which stands for symbiotic culture of bacteria and yeast, but it would be more correctly referred to as the pellicle, which is the cellulose floating biofilm or mass that is created mostly through the action of Bacter xylanus, which is a gram-negative arrow. The yeast in the culture produce invertase to separate the disaccharide sucrose into glucose and fructose, which is then absorbed by the K-xylanus and made into cellulose and excreted through pores in the cell membrane. These then aggregate together to form this mat. Biofilms are usually studied for their formation on solid surfaces. Every time you brush your teeth, you are fighting the formation of a biofilm on the surface of your teeth. And any surface that is periodically suffused with water and nutrients can grow biofilms. And biofilms are formed by bacteria to increase their odds of survival. It allows them to better resist changes in pH. They can resist antibiotics or changes in osmolarity. This is sort of an unusual case because it's forming on the surface of a liquid rather than the surface of a solid. This product can be applied to several different things. First, the textile applications. It can be pressed or dried into leather-like material. And this image shows a pellicle that is still wet. And you can see how it is translucent because it can hold up to 200 times its dry mass in water. This one is dry, and you can see that it looks a lot more like leather. There can be problems that arise with using it for textiles because of this property, because it can draw water from the air, being hygroscopic, and then this can be a environment where mold could grow. So if you want to use it for textiles, you have to take steps to prevent that. And this is really interesting because it can be used to replace less environmentally friendly textiles, and it can be formed using waste materials that would otherwise be thrown away. It can be used for food applications. In this image, this spiral on top of the pie is using bacterial cellulose from kombucha mixed with fruit to make a fruit roll-up like food. And in food, it can be used as a gelling agent similar to pectin. And this can also be another way that food waste can be diverted, and it has potential for creating low-calorie foods since it increases the sensation of satiety. It also could be used for biodegradable food packaging, which would divert a lot of plastic waste. In the biomedical field, there's a lot of interesting applications. In these cases, they use never been dried microbial cellulose, so it hasn't lost its unique nanocrystalline structure of cellulose, but it has been treated with a strong base so that all of the bacterial cells will be removed so as to not cause an immune response in the patient. It can be used for the delivery of medicine, and it has been shown in small studies to perform better than conventional wound dressings for burns. It can be augmented during its production or with therapeutic agents that are designed to facilitate wound healing. It also has been shown to reduce pain when applied to burns. it can be used to make highly biocompatible, long-lasting implants that will not be attacked by the host immune system, and they can act as a scaffold for cells to grow into, and it has been used in animal studies to create man-made blood vessels. In my experiment, I brewed 3 batches with varying concentrations of sucrose to see what the effect on the durability of the resulting pellicles would be. The first group had no sugar except that which was contained in the inoculant. The inoculant in this case was the commercially available Synergy GT kombucha, and that's for the sake of replicability rather than using a culture that only I have, since cultures vary a lot from batch to batch or even over the course of fermentation or regionally. And sucrose has been shown to have the highest production of bacterial cellulose over periods longer than 84 hours. It's the lowest production in the first 84 hours because the sucrose has to be hydrolyzed into glucose and fructose in the periplasmic space before it can enter the cells and be used for fuel. Sucrose is also thought to increase how the cellulose nanofibrils are interwoven with each other. In this image, they were all grown in these bottles over a period of 3 weeks. This is from group 3 when they were still wet, and this is group 3 when they were dried. For the data collection portion, I used a force transducer to measure the maximum amount of force before the pellicles could rip, and the maximum force was measured for each pellicle. In the first group and the second group, the average things that they withstood weren't very different. But going to the third group with the highest sucrose concentration, there was a much higher amount of newtons that could be withstood before it tore. And there was a statistically significant difference between these groups. As for the microscopy, there aren't a lot of differences that could be observed at 400 times magnification, but the thinnest pellicle was the most interesting to look at because it had these dark striations that were caused by wrinkling from the pellicle drying unevenly and contracting in certain areas more than others. And these wrinkles were visible under the microscope, and larger wrinkles were visible with the naked eye. And there's also this cellular structure that appears to be the remnants of dead yeast cells that have been incorporated into the pellicle. In the other 2 groups, since they were thicker, there was a lot more refraction and it was harder to get a clear picture on them. These are some of my works cited, and I got some questions on my poster presentation, which I wanted to answer here. One is there any condition which would allow for homogeneous growth of the pellicle? And that's really interesting because a lot of the studies that I looked at did create more homogeneous pellicles because they isolated the K. xylanus strain. That's what this one is on, even though it says Gluconeacetobacter xylanus. That's the previously used name of K. xylanus. And since they were an isolated species, of course the product is much more homogeneous and lacked the yeast cells which are visible in these images. And then the same person also asked, there was a small difference between the first 2 groups, but then a very large difference in the third group. And do I believe that it was caused by a certain threshold of sucrose being needed? And that is a good question. I'm not sure if there's a threshold value, but something that would have been interesting to look at would have been if the first and second group would have continued to grow if they had just not reached their peak yet, because there were less resources available, so their lagging phase could have been longer. The, um, this same study looked at the different incubation times and how that affected it. So that would be a good thing to look at if you wanted to answer that question in more detail. Thank you so much, and have a good rest of your day.